Today on Yesyouchem, we'll be talking about absorbance, fluorescence spectroscopy, plus
we'll be talking to Dr. Vincent Helser about how he's using spectroscopy to analyze proteins. Spectroscopy. What is spectroscopy? Well, spectroscopy at its root would be the interaction between electromagnetic radiation and any type of molecule that radiation encounters. But in science, there's two fields of spectroscopy: analytical spectroscopy and physical spectroscopy. In analytical spectroscopy, we use this electromagnetic radiation to measure the amount or the concentration of any solution. In physical spectroscopy, we use this radiation to really analyze the structure of the molecule and any physical properties that molecule may have. And when we talk about analytical spectroscopy, we'll most likely be dealing with absorbance. What is absorbance? Redundantly put, absorbance is the absorption of any photon energy. Let's talk about the subject in more detail by starting with the hydrogen atom. Now here we have the hydrogen atom with one electron in ground state configuration. Now it takes a particular amount of energy for this electron in ground state configuration to move to a higher energy orbital. You'll notice there are 6 lines. Each line represents a different energy orbital, 1 being the lowest, 6 being the highest. It takes 122 nanometers of light for this electron to move to the n equals 2 orbital. But it can also move to the n equals 3 from n equals 1. And vice versa. It just takes a shorter wavelength of light, which is more energy, to move it there. Now, let's start from a different energy orbital. Instead of n equals 1, we'll start from n equals 2. Now this transfer took 656 nanometers wavelength of light. That is less energy, a longer wavelength. You can go from n equals 2 to n equals 4, n equals 5, and n equals 6. Now we don't have to start from n equals 2 either. We can start from n equals 3, which is a higher energy. And it takes even longer wavelengths of light, less energy, to move this electron up in energy. Now, here's the absorbance spectrum of light. It's sort of a fingerprint for the hydrogen atom. You'll notice the black bands here are wavelengths of light that the hydrogen atom absorbed, and you can't see anything. If we transfer this Balmer series over, you'll see that it perfectly matches up. The Balmer series, in this case, is the only absorbance spectra that we can observe because it's visible light. But the Lyman series that we discussed in the first place is observed in the UV spectrum, and it takes more than just the human eye to observe it. The Paston series is observed in the IR light. Every element has its own absorption spectrum, its own fingerprint, if you will. We want to stress that it takes a certain amount of energy for an electron to be promoted to a specific orbital, just like it takes a proper amount of force for you to make a perfect swish in basketball. So how do we use absorbance? Absorbance spectroscopy takes into account how much light passes through this molecule or through this atom, because it's really hard to measure how much exactly the electron itself absorbs. So if we take the transmittance of light through the absorbance, you'll note that transmittance is logarithmically related to absorbance.
Yo dude, beer tonight?
Like beer? No. Beer's law?
Yeah.
So Beer's law, absorbance equals extinction coefficient times concentration times path length. Let's go first over the units and why this formula works. The extinction coefficient is taken as liters per mole inverse centimeters inverse. You have molarity as your standard concentration measurement, moles over liters, and then you have the standard centimeter path length. Now, how do these cancel out? Mole inverse, we'll deal with this, centimeters also, and then you have liters over here, which crossed out, which leads to an absorbance that has no units. That's important to note. Absorbance never has any units. Now, extinction coefficient is something that we use, essentially, to measure out how strong a molecule absorbs light at a particular wavelength. So you'll always see extinction coefficients like this. You'll see epsilon, and then you'll see a particular wavelength that we are questioning, 280 nanometers. So another thing to note is that the standard for this formula is a 1 centimeter path length, but you might not always have a 1 centimeter path length. You might have a 1 millimeter path length. Why would you have a 1 millimeter path length? That always happens whenever you are trying to measure out the absorbance of a very, very, very dilute solution. You would get a smaller cuvette with a smaller path length. Those are the basic concepts behind Beer's law.
Now let's do an absorbance practice problem. We've been given this graph of absorbance versus wavelength, and the peak absorbance is somewhere around 0.25. That's where the value comes from below. For the first part, we've been given the absorbance of 0.25, a path length of 1 millimeter, and an extinction coefficient of 116,000 per centimeter molar. You should recognize that what we need to solve for the missing quantity, concentration, is Beer's law. Let's plug in what we have. You'll see that absorbance is a unitless quantity, but on the right side these units don't cancel. Molar cancels, but then we're left with centimeters and millimeters. All we have to do is convert from millimeters to centimeters. There are 10 millimeters in every centimeter. Finally, let's simplify and complete this algebra to find that the concentration is 2.2 times 10 to the negative fifth molar.
So what is fluorescence?
Fluorescence, the light emitted by an atom or molecule after a finite duration subsequent to the absorption of electromagnetic energy. So there's 3 steps to it. First, an electron is going to be at a ground state. It's going to be excited by photons. There will be a loss of energy and an eventual return down to the ground state. Now, usually, when it returns down to the ground state, it will only release thermal energy. However, in a select few molecules called fluorophores, it will either emit visible light, infrared light, or UV light. The light in this case blue light, must be of a shorter wavelength and therefore a higher energy than the light emitted, simply because there is a loss of energy when you have to promote the electron up. The example here is green fluorescent protein, or GFP. This is used in many wet labs, usually to tag cells for stuff like confocal microscopy or any type of live imaging. It can also be used in live models such as rats to track tumors. It's used in flow cytometry for cell sorting. It's basically extremely applicable.
Let's take green fluorescent protein, for example. It's excited by blue light, moves up to an excited energy state, then loses a little bit of thermal energy by vibrating, and then jumps all the way back down to ground state and fluoresces green light. Some molecules don't fluoresce at all. Upon absorbing wavelengths of light, the electrons get excited, but on the way back down, instead of fluorescing light, they release thermal energy.
I'm Vince Hilser. I'm the chair of the biology department, and I'm a professor in the department as well. Biology is a discipline that's very broad, and I deal with the molecular mechanisms of how protein molecules work. In particular, I study how information, so we know that proteins are not just structures, static structures, they sort of undergo conformational changes as part of their function. How they go about these movements and the implications of how binding drugs or binding their receptors affects other parts of the molecule, which allows them to interact with other things. That's what we try and study, because it's at the heart of really all biology. Mostly what we use is spectroscopic methods, and they can be NMR spectroscopy or different forms of light spectroscopy. Things like circular dichroism and fluorescence and ultraviolet and visible spectroscopy. We have to take advantage of the properties of these molecules. And so molecules are made up of protons and neutrons and electrons, and that allows us then to perturb them with energy. And the energy that we perturb them with is light. In order to see them and see certain aspects and properties of them, we use spectroscopy. So depending on what we want to see, we employ that type of spectroscopy. So it's really, spectroscopy is the key to us being able to observe them. One particular spectroscopy that we use is circular dichroism. For instance, alpha helices, a structure that's that's common in proteins, has a certain characteristic absorption property with polarized light. So we can look to see how structured molecules are under certain conditions. So it's just a general technique that allows us to understand whether it's structured, and then if you look at that property as a function of adding another molecule that it binds, or destabilizing it with temperature, or seeing what effect lowering the pH has on it, you can see that change in that property with those conditions. And over the years, the field has learned how to analyze that information and determine a lot about it. How stable is it? Does a mutation to this as a result of a disease? Does that destabilize? What's the origin of the disease? We get at the molecular basis of the disease, and that molecular basis is usually when something happens. There's a difference, a genetic mutation that will affect a physical property of the protein. We're interested in a class of molecules called transcription factors, and these are things that turn genes on and off. The class of transcription factors we're looking at are the class called steroid hormone receptors, and they're important because in that family of proteins is estrogen receptor, and glucocorticoid receptor, and progesterone receptor, and androgen receptor. These are all, if they don't ring a bell, they should ring a bell, and that what they are involved in is significant amounts of cancers. Androgen receptor is prostate cancer, and estrogen receptor is breast cancer. And misregulation or dysregulation of these molecules is associated with these types of cancers. So we have to know how these things work, and how the mutations or how whatever is going wrong with these is related to cancer. So this is what we're studying right now, and it turns out there's a whole class of protein behavior that has been discovered in the last 20 years, and that's these sort of sequences that are what are called intrinsically disordered. That is, the fluctuations that I study that we sort of allude to in biology, where you have structured molecules that sort of move around a little bit around an average structure. That's important. But these kind of molecules, they have no unique conformation. They're basically flopping all over the place. So they're really difficult to study. But to understand why people get cancer, we have to understand that process. So we've taken all of these techniques, spectroscopy being forefront among them, to go in there and interrogate these systems and figure out how we're going to make a difference at a medically relevant level.
Thank you, Dr. Hilser. When it comes to beating cancer, you need spectroscopy. If you ever plan to research prostate cancer, breast cancer, you're going to be analyzing micromolecules responsible for those respective conditions. And we couldn't really do that without spectroscopy.
Just like Dr. Hilser said, research relies on observations at this microscopic level. Spectroscopy opens up the world of these biomolecular molecules, such as the proteins that he was analyzing, to really solve cancer.
Guys, thank you for joining us today. And remember, with us,
Yes, you can.